in both their structure and their function. So as we take a look at these different cells, they differ in both structure as well as their function. And the cell diversity that's associated with the 200 different types of cells that make up the trillions of cells that make up our human body give rise to a distribution of labor, in which based on cell structure and function, different cells will carry out different functions. We know that muscle cells are equipped for that of movement. Our red blood cells are equipped for transporting oxygen throughout the cells of the body. Nerve cells are responsible for generating electrical signals, which are then called release of neurotransmitters, which will regulate cellular communication. So because we have these 200 different types of cells that differ in both their structure and function, distribution of labor, which allows the body to carry out multiple functions to maintain homeostasis. But when we take a look at the cell diversity, we can take a look at these cells in general. So this is just in general. And generally speaking, we can say that despite the diversity of the 200 different types of cells that make our body up, there are some basic components associated with these cells. So we look at the general components or basic components associated with most of the cells of our body. Most cells or most eukaryotic cells of the human body will consist of major components. So we can highlight three major components. And the three major components that are associated with virtually most of the cells in our body. Now there are some exceptions to the rule, but the three major components, we have that of our what? Cytoplasm, component. We have the nucleus, and then we have that of the plasma membrane. So generally speaking, when we take a look at the basic components of the eukaryotic cells that make up the human body, these cells will consist of three basic components despite the diversity, and this is the cytoplasm, the nucleus, and the plasma membrane. Now we do know there are certain cells that are anucleated, for example that of our what? Red blood cells lack a nucleus, but generally speaking we have three major components associated with the cells of our body. And as we take a look at the cytoplasm, how can we describe the cytoplasm of a cell? It's just going to be what? All that material that surrounds the nucleus and is enclosed by the plasma membrane. It's all the material that surrounds the nucleus, this intranucleus, and it's enclosed by the plasma membrane, PM for plasma membrane. So our working definition for the cytoplasm will be all the material that surrounds the nucleus of the cell and is bound by the barriers established by that of the plasma membrane. Two components, and the two components of the cytoplasm of these cells will be that of our what? And the what? Organelles. So we have the two major components. We have the cytosol, and we have our organelles. And when we take a quick look at the cytosol of the cytoplasm of the cell, cytosol will be that of the intracellular fluid. So it's a semi-fluid-like material that's enclosed by the plasma membrane. So it's the intracellular fluid. It's a semi-fluid-like material that consists of water, primarily water, and the dissolved solids within the cell, the nutrients that allow the cell to carry out its function. It consists primarily of water plus our dissolved solids. But when we think of the cytosol of the cell, we also want to highlight that the cytosol is the site of action. cellular activity will occur within inside the cytosol of the cell. For example, we know that we have ribosomes suspended within the cytosol of the cell. And we know our ribosomes function is that of what? Protein synthesis. So this will be a site of protein synthesis. So this will be a site of protein synthesis within inside the cytosol of the cell. Also, metabolic reactions occur within inside the cell. Chemical reactions occur within of the cell. So we want to keep in mind this is a hot spot of the cell. So we have activity occurring within the cytosol. We think about generating energy from nutrient molecules. We know that glycolysis occurs in the what? Cytosol. Then we have the electron transport chain occurring within the what? Mitochondria. But we have metabolic activity such as that of glycolysis occurring within inside the cytosol or the liquid of the cytoplasm of the cell. Then we have the organelles component of the cytoplasm, and we can describe our organelles as just being specialized structures or specialized intracellular structures that have specific what? Function. So they're just intracellular structures that have a specific function. And as you take a look at the organelles suspended within inside the cytosol of the cytoplasm cell, we can differentiate the organelles as either being membranous organelles or non-membranous. So we have them classified as being membranous organelles or non-membranous. For example, the non-membranous structures associated with the cell will include that of our ribosomes. These are non-membranous structures. We have cytoskeleton components. So we have the ribosomes, cytoskeleton elements, centrioles associated with the eukaryotic cells in the human body. These would be some of the non-membranous organelles. Make sure you know the function of the organelles of the cell. Then we think about membranous organelles. We have our Golgi apparatus. We have the endoplasmic reticulum. We have that of our lysosomes, peroxisomes, the mitochondria. For example, this is the mitochondria. And what's the advantage of having membranous organelles inside the cell? What's the advantage of the cell for having membranous structures? What does this allow the cell to do? Somebody. Transport. What do you mean when you say transport?
Like if you pinch off vesicles.
All right, so pinch off vesicles. Give me another. What's the general major function of our cells being associated with membranous organelles? What would the book tell us? What does it allow the cell? Exactly. It allows us to compartmentalize certain activities within certain regions of the cell. We have different environments. We think about that. Overall pH in the cytoplasm of the cell is roughly 7.2. Lysosomes, hydrolytic enzymes, they have an environment roughly of 2.0. So therefore allows us to compartmentalize activities within the cell, have discrete environments within the cell to carry out particular functions. We want you to make sure you take a look at the table in our textbook. I believe it's table 3.3, where they highlight. Make sure you take a look at the table. I could be wrong, but I think it's 3.3. Make sure you look at the table that highlights the function and structure of our organelles. Now, I could be wrong on the number of the table, but the point I'm trying to get across here is make sure you take a look at the table in the textbook that highlights both the structure and the function. Do we see that in the book? Is it 3.3? Okay, excellent. Make sure you take a look at the table that highlights the structure and function of the organelles, and from here on out, throughout the entire semester, you'll be responsible for knowing the function and the structure of all the organelles associated with the cells of the human body. Then we take a look at the nucleus, and as we look at the cell's nucleus, we commonly refer to the nucleus of the cell as being the what? Control center, or the brain. You might see the brain, but we commonly refer to it as being the control center of the cell, and we call the nucleus the control center of the cell. Why do we call it that? Because it houses what? Most of the what? Cellular DNA. So the nucleus will be associated with approximately 99% of the cellular DNA, and by housing most of the DNA of the cell, it will be able to what? Direct the synthesis of the what? Proteins that the cell makes. So it will direct, so we call it the control center because it houses the genetic material of the cell, and it will direct the synthesis of our proteins. When we think about our proteins, the proteins of the cell play a major role in determining the what? Structure and the what? Function of the cell. So the nucleus is the control center of the cell because it directs the synthesis of the proteins that the cell will make, and by directing the synthesis of the proteins, we determine the structure and the function of the cell. We think about most of the cells of the human body have the same genome. When we say genome, they have the same what? Material, but not all those genes will be turned on in every cell. They specialize in what's called the what? Proteome. So it's the proteome. Elizabeth, when we say proteome, what do you mean when we say proteome?
The whole genetic makeup of protein, like the protein, all like how you have the genome, all the genes, and you have the proteins, all the.
You say proteome, Daniel. What's it mean specifically? Exactly, so it's a type and the number of proteins that a cell makes, and it's the proteome of a cell that will determine whether it's a what? A muscle cell, a nerve cell, a red blood cell. These cells are different in structure because they differ in their what? Proteome, because those cells will turn on certain genes for certain proteins that won't be turned on in other cells. The proteome that gives rise to the cell diversity. Do we get that point across? So we're trying to say that's the key to what determines whether it will be a cell, nerve cell, or epithelial cell. So we have the nucleus. Make sure you're familiar with the structure of the nucleus, being enclosed by the nuclear envelope, communicating with the cytoplasm by way of the nuclear pores, bringing information into and out of the nucleus. Then we have the plasma membrane, the other major component of the cell, and we know that our plasma membrane encloses each individual cell will be enclosed by a plasma membrane. So it encloses each cell. The plasma membrane will serve to establish what? Boundaries between the intracellular fluid and the extracellular fluid, or the interstitial fluid. So it establishes boundaries. A plasma membrane will also regulate the exchange of material between the intracellular fluid and the extracellular fluid. So it regulates the exchange of material between what's inside the cell, intracellular fluid, and the intercellular fluid. So we're taking a look at the general structure of a typical cell of the human body, the three major components: cytoplasm with the cytosol and the organelles. You are responsible for this table to make sure you know the function of all the organelles. A nucleus, we're saying that's a control center because it has most of the DNA. 99% is housed within the nucleus. Where's the remainder of the cellular DNA? In the mitochondria, so it has its genes that code for mitochondrial proteins that allows the mitochondria to carry out its function. And then we have the plasma membrane. So we want to take a quick couple moments to take a look in a little bit more detail. Membrane, boundaries, regulating the exchange of material between intracellular and fluid. What are the four major components or molecular components of the plasma membrane? What type of molecules make up the plasma membrane? We have proteins, carbohydrates, lipids, and cholesterol. So we look at the four major components of the plasma membrane. That would be our phospholipids, our cholesterol, the proteins, and the carbohydrates. So these are the four major components of the plasma membrane: phospholipids, cholesterol, proteins, and the carbohydrates. And we take a look at the structure and function of these four major components of the plasma membrane. We see here we have the phospholipids, the carbohydrates highlighted in yellow, proteins highlighted in the lavender or the purple, the green represents the carbohydrates. And as we take a look, the phospholipids can be viewed as the major structural component of the plasma membrane, so they make up the major structural component of the plasma membrane. Will be that of the phospholipids. We take a look at the general structure of the phospholipids. Structurally, they will consist of a molecule of glycerol. So we draw a cartoon-like structure of the phospholipids. We have a molecule of glycerol covalently attached to a phosphate group, and the phosphate group will be attached to a nitrogen-containing molecule called choline. And then also attached to the glycerol molecule of the phospholipid, we will have what? Two fatty acids, or two fatty acid tails. So we have fatty acids. Structurally, we know that our fatty acids are what? Long hydrocarbons that have a what? Carboxyl group. So structurally, long hydrocarbons. General structure, we can say CH3, CH2, N. N just represents how many CH2 groups we have. At the end, we'll have a carboxyl group. So this would be the general structure of the fatty acids. If we take a look at the fatty acids, we look at this depiction here. We have this fatty acid having a small beta fatty acids. Many of the fatty acids associated with the structure of our phospholipids are said to be unsaturated fatty acids, and when we say they're unsaturated fatty acids associated with the glycerol molecule, which forms the phospholipid, what do we mean when we say they're unsaturated fatty acids?
One or more carbon to carbon.
Exactly. So when we think about them unsaturated, they have at least one or more carbon to carbon double bond. And what does that carbon to carbon double bond do to that hydrocarbon? It will put a kink or a what? Bend in the fatty acid tail. So our carbon to carbon double bond will put a kink or a bend, so to speak, in the fatty acid tail. And by bending or causing a kink in the fatty acid tail, effectively, what does this do to the phospholipid and the plasma membrane in general? What physical property does it afford to the plasma membrane? Say it. Well, the fatty acids make it hydrophobic on the core. About this unsaturated fatty acids, the kink in the fatty acid tail will increase the distance between adjacent phospholipids. So the fatty acid tail will increase the distance between adjacent fatty acids. So it increases the distance between adjacent fatty acids, and by increasing the distance between adjacent fatty acids, what it will do effectively is it will decrease van der Waals interaction. Van der Waals interactions are weak attractions between transient charges between atoms. So it will decrease van der Waals interactions, which are just weak attractions between opposite charges. And by decreasing the van der Waals interactions, by decreasing the distance between adjacent fatty acid tails, are we okay with what we're trying to say here? We know positive to what? A negative charge. And so by increasing the distance between transient positive and transient negative charge, we decrease van der Waals interactions, we prevent the crystallization of our fatty acids, and this allows the plasma membrane to be fluid-like in nature. So by having a large percentage of the fatty acid tails associated with the phospholipids being unsaturated, carbon-to-carbon double bond, increasing the distance between the different fatty acid tails, we decrease van der Waals interactions, we prevent the crystallization of the fatty acids, and therefore this helps to maintain the fluidity of our plasma membrane. I know we've heard the term the fluid mosaic model. So the plasma membrane is not a rigid structure, it's a fluid-like structure. Is that a question right here?
Yeah. Do all the phospholipids, do they all contain unsaturated and saturated? Does all of them?
Not all of them. And we have a large percentage that will have an unsaturated fatty acid. What's that doing is increase the distance between adjacent fatty acid tails, which helps to regulate the fluidity or maintain the fluidity of the plasma membrane. But I mean, if you take a look at the picture, they're not highlighting that. It's a concept we're trying to get. So as we take a look, this helps to maintain the fluidity of the plasma membrane. Now we know when we take a look at the general structure of the phospholipids chemically are referred to as what? Amphipathic. So they're amphipathic molecules. And what do we mean when we say amphipathic? They have both a what? Hydrophobic and a hydrophilic region. The phosphate head will be the hydrophilic, which can readily interact with water. We know our fatty acid tails being hydrocarbon primarily will be the hydrophobic region. And based on the amphipathic nature of the phospholipids, when they're immersed in water, phospholipids will self-polymerize or self-assemble would be a better term, and they will form the lipid bilayer, which is the general structure of the plasma membrane. So based on the amphipathic nature of the phospholipids and the hydrophilic region associated with the phosphate head and the hydrophobic region associated with the fatty acid tails, when they're submerged in water, the phospholipids will self-assemble and they will form the lipid bilayer, which is the general structure of the plasma membrane. So they will assemble the plasma layer. And the lipid bilayer is approximately 10 nanometers in thickness. At the hydrophilic region exposed to one compartment of water, this can be the intracellular fluid, and the other side of the plasma membrane will be exposed to the other component of water, will be the extracellular fluid, and the fatty acid tails, which are hydrophobic, will sequester themselves away from what? Water. And as they sequester themselves away from water, our plasma membrane will be associated with a hydrophobic core. A hydrophobic core made of fatty acid tails associated with the phospholipid. And what physical property does this hydrophobic core afford to the plasma membrane? What physical property does it give to our plasma membrane? It makes our plasma membrane what? Selectively permeable. So we know a term used to describe the plasma membrane is semi-permeable or selectively permeable. So because of the hydrophobic core formed by the sequestration of the fatty acid tails away from the intracellular fluid and the extracellular fluid, structurally we have a hydrophobic core which allows the plasma to regulate what can readily enter and exit the cell. The plasma membrane is readily permeable to lipophilic substances. So our hydrophobic molecules can readily, or non-polar molecules can readily enter and exit the plasma membrane. So lipids can gain access directly into the cell. This also fat soluble vitamins such as vitamin A, D, E, and K. Because they're fat soluble, they can readily interact with the core of the plasma membrane. The plasma membrane is also go to our gases, such as our O2 and our CO2, and it's also somewhat permeable to small molecules. Even though water is a polar molecule, to the small size, it's also permeable to small molecules. For example, such that as water. So despite the polar nature of water, because it's a relatively small molecule, it can readily interact or cross the plasma membrane. The plasma membrane is impermeable to large, polar, and charged molecules. So it's impermeable to large, polar, and charged molecules, and therefore it's impermeable to molecules such as our glucose, our ions, our amino acids, which give rise to our proteins. And we know that we have to get glucose into the cell. Ions have to be able to move into and out of the cell. And because we're saying they're impermeable to the plasma membrane, how can we get this essential material into the cell?
Next to charge.
So how can we get these materials into the cell? Exactly. So despite the fact that they cannot readily enter into the cell, we use the assistance of transport proteins to get material into the cell, which is said to be impermeable to the plasma membrane or cannot readily enter or interact with the hydrophobic core formed by our fatty acid tail. Take a look at the phospholipids, key structure component, but also due to the hydrophobic nature or amphipathic nature, they form the hydrophobic core, which will regulate the interaction of material into and out of the cell. We want to take a look at the remaining components of the plasma membrane, and we have that of the cholesterol. And as we take a look at cholesterol and its role with regards to plasma membrane, take a look here. We're highlighting the cholesterol molecule associated with the plasma membrane. Take a look at the structure of cholesterol. Cholesterol is a large, bulky molecule. It's mainly hydrophilic, but it is weakly hydrophilic. The molecule is weakly amphipathic. You take a look at our cholesterol. It's a large, bulky molecule. Weakly amphipathic, mainly hydrophobic, but we take a look, we have a hydroxyl group associated with one of our 6-string carbons, and this hydroxyl group, cholesterol molecule, weakly amphipathic. So the hydroxyl associated with the structure of cholesterol makes this molecule weakly amphipathic. And as we take a look at the role of cholesterol, it will help to regulate the fluidity of the plasma membrane. So its major role is to regulate the fluidity of the plasma membrane. We don't want the plasma membrane to become too fluid-like or too rich. So it helps to regulate the fluidity of the plasma membrane. Case in point, if we were to have an increase in body temperature, an increase in body temperature will be associated with an increase in what? Molecular movement. So as you increase body temperature, increase the molecular movement. And as we increase molecular movement, the hydrophilic portion of the cholesterol molecule in the hydroxyl group can limit the lateral movement of the phospholipids on either side of the plasma membrane. So at increased temperature, increased molecular movement, it can limit the movement, inframovement of the phospholipids, preventing the plasma membrane from being too fluid-like. If the plasma membrane is too fluid-like, what can happen? If it becomes too fluid-like, we can what? Compromise the integrity of the what? Plasma membrane, and then the extracellular fluid can leak right into the what? Intracellular fluid. Does that make any sense? So we're saying it can limit the lateral movement of the phospholipids by interacting with the phospholipids by way of this hydroxyl group. The converse, if we decrease body temperature, conversely, we will have a decrease in molecular movement. The phobic portion, which is the major portion of the molecule, will function as a molecular spacer between adjacent phospholipids. So the decreased movement, it can act as a molecular so to speak. The hydrophobic portion increase the distance between adjacent fatty acids, so it can act as a molecular spacer and increase the distance between adjacent fatty acids. And as we increase the distance between adjacent fatty acids, effectively, what are we doing?